What we have done here is to take these antibodies and combine them with chemotherapy drugs and show that they can be very effective. Now, when people make nanoparticles, they usually talk about spheres. They make a nice spherical round particle, and that's good. But what we found out and what we hypothesized is that if you make the particles into a different shape, in our case a rod, they work much better.
The major problem with the current anti-cancer drugs is to deliver the correct amount of the drugs to the right cells. We prepared nanoparticles of rod and disc-shaped particles from spherical nanoparticles, and we coated the surfaces of these nanoparticles with a monoclonal antibody which is known as trastuzumab. Trastuzumab binds on the cancer cell surface with a certain receptor proteins which is known as HER2 receptor proteins. When the antibody binds with the HER2 receptor proteins, it stops the activity of HER2 and thus stops the cancer cell growth. But the problem with the trastuzumab, its efficiency is very low. And we found that using the rod-shaped nanoparticles, we could increase the therapeutic efficiency up to 50% using only 1 microgram per ml of the trastuzumab solution.
So imagine this nanoparticle moving through the bloodstream and finding a cancer cell. When it finds the cell, it wants to bind to it, and the sphere is not able to have that much contact area between itself and the cell surface. In contrast, if you have a rod, you can imagine it sitting on its side and making a much better contact with the surface of the cell.
Presenting the antibody on the different shapes of nanoparticles, we not only enhances the interactions with the breast cancer cell surface, we also inhibited the cancer cell growth using very tiny doses of antibody, which is not toxic to the healthy cells. In the next step, we replace the polystyrene particles with a pure drug nanoparticles. For that, we chose a model drug which is chemotherapy, and chemotherapy is an anti-cancer drug which is in phase 1 clinical trial. When we loaded the trastuzumab antibody-coated chemotherapy nanorods to breast cancer cells, we found that the 50% growth inhibition of breast cancer cells can be obtained by using very tiny doses of chemotherapy.
We think that our findings will open up new opportunities in terms of making particles very selective and more effective against a variety of diseases, not just cancer, but any other target. We think we can use this idea developed here to make them work much better.